The cell's nuclear envelope contains approximately 2,000 nuclear pore complexes that allow the transport of water-soluble molecules such as RNA, ribosomes, certain proteins, carbohydrates, and signal molecules. By adding labeled cargo molecules to permeabilized cells, the movement of single molecules through the nuclear pore can be visualized by fluorescence microscopy. Time-lapse images are required to determine localization, as well as spatial temporal characteristics such as transport time, import-export efficiency, and entrance frequency.
Hi, I'm Goryaynov, PhD student from the Laboratory of Single Molecule Microscopy in the Department of Biology at the Bowling Green State University.
I'm Ashapurna Sarma, a master's student, also from the Single Molecule Microscopy Lab.
I'm Jiong. I'm a postdoc fellow here at Single Molecular Microscopy Lab.
Today, we will show you a procedure of single molecule imaging of nuclear transport through the nuclear pore complexes in the helo cells.
We use this procedure in our lab to study the translocation of various molecules through nuclear pore complex.
So, let's get started.
To prepare the proteins for the nuclear transport experiment, conventionally transformed JM109 E. coli, expressing the genes responsible for production of the nuclear cargo proteins, are grown in LB media and induced with IPTG. Once the cells are in log phase with an optical density of 0.7 to 0.8, spin the culture at 6,000 times gravity.